Welcome to Ramya Teacher's Classroom. Today, we're bringing the laboratory right to your screen with a really exciting biology explainer. Have you ever stopped to wonder what actually happens the exact moment food hits your tongue? Well, today we're exploring a classic foundational experiment that reveals the invisible, almost magical chemistry happening in your mouth every single time you eat. We are looking at the salivary amylase reaction on starch digestion. So here's our quick roadmap for today's experiment. First up, we'll meet the chemical cast, then setting up the experiment. After that, we get to the iodine test reveal, and finally, we wrap it all up with the grand biological conclusion. Okay, let's dive into this. Section 1. Meet the chemical cast. Our very first player in this lineup is starch. Now, in the context of this experiment, starch is what we call our substrate. You can just think of a substrate as the raw material that's about to get worked on. It's the primary carbohydrate we're observing here, literally the exact same complex carb you'd find in a bowl of rice, a potato, or a slice of bread. Next up, we have our active worker, salivary amylase, also known as ptyalin. Now, this is an enzyme which basically acts like a tiny biological machine. And the truly amazing part, it's found naturally right inside human saliva. This is the catalyst that's going to interact directly with our starch substrate. And our third key term for today is maltose. Maltose is a type of sugar, and for us, it's the final product of this experiment. See, when our enzyme worker, that salivary amylase, gets a hold of the starch substrate, it goes right to work breaking it down. The finished, newly processed result of all that breakdown, that's maltose. All right, moving right along to section 2, setting up the experiment. Now, let's look closely at the very foundation of our scientific method here. We've got two identical test tubes, test tube A and test tube B. To test tube A, we're adding saliva, but to test tube B, we just add water. Why? Well, test tube B acts as our scientific control. Without this water-only control, we literally wouldn't be able to conclusively prove that the saliva itself is causing the chemical changes we're about to see. It's absolutely crucial for comparison. Of course, to maintain a perfectly valid experimental environment, we've got to follow some exact procedural rules. First off, we take equal quantities of our starch solution in both test tubes. Balance is key here. Second, we have to maintain warm conditions, so we'd usually use a gentle water bath. But notice the strict warning on this one. Do not use boiling water. That would ruin the enzymes. And finally, we absolutely must keep those test tubes perfectly clean to avoid any outside contamination that could mess up our data. Let's move to and see how this builds. Section 3, the iodine test reveal. So this brings up a classic viva voce, an oral exam question for the class. Why exactly do we use iodine in this experiment? Right now, both of our test tubes just contain these clear or kind of cloudy liquids. So how on earth do we detect invisible chemical changes that are happening on a microscopic level? Well, the answer is that iodine is basically our chemical detective. We use it specifically to test for the presence of starch. When iodine comes into contact with starch, this dramatic visual change happens. It instantly turns a deep, dark blue-black color. So if we see blue-black, we know for a fact that starch is definitely still hanging around in that tube. But just like with our initial setup, there are strict rules for handling our detective chemical here. You must use separate droppers for the saliva and the iodine solution to prevent any cross-contamination. And this is super crucial. You have to observe the color in the test tubes immediately after adding the iodine to get an accurate reading. And this brilliantly illustrates our results. Just look at that visual contrast. After warming the tubes in our water bath and adding those iodine drops, test tube A has turned a brown color. But test tube B, our handy control tube, has turned a deep, inky blue-black. Which brings us to section 4, the grand biological conclusion. Let's decode this evidence together, shall we? Test tube B, our control tube with just water, turned blue-black. And as we just established, that means the starch is still perfectly present and totally untouched. But test tube A, the one we treated with saliva, it turned brown instead of blue-black. Because it didn't give us that blue-black reaction, it means the starch has completely disappeared from the solution. There's no starch left. So the crucial point is this. The salivary amylase inside the saliva successfully worked on the starch. It actually broke down our starting substrate and turned it right into maltose sugar.
Quite literally, the saliva digested the starch right there in the test tube. How cool is that?
Which leads us straight to the ultimate biological inference of this entire explainer. Digestion of carbohydrates begins in the mouth. It doesn't start in your stomach like a lot of people think. The exact moment you chew your food and mix it up with your saliva, that ptyalin enzyme goes straight to work, breaking down those complex starches into simpler sugars before you even swallow.
Which leaves us with something pretty fascinating to think about for our own daily lives. If chewing is the mechanical process that mixes your food with this incredible salivary amylase, how does eating way too quickly and not chewing your food enough affect your body's ability to digest carbohydrates? Seriously, the next time you grab a quick bite, remember the literal chemistry happening right on your tongue. Thank you so much for joining me for this explainer, and keep exploring the incredible science happening all around and inside you.